All right, so we are on our last lesson for Unit 5. Today we're going to be doing DNA technology. What we're going to learn about is interpreting DNA, comparing and identifying the organism, summarizing transgenic organisms engineered to benefit society, as well as elevate some ethical issues surrounding the use of DNA technology like cloning, GMOs, STEM research, and Human Genome Project. See what your thoughts are. But how do we use DNA? We know that DNA is here, but how do we actually use it? Well, DNA can be used for comparison and identification, right? DNA is found in every part of your body. It's found in your hair cells, it's found in your body cells, it's found in your toenails, it's found in your eyelash. Um, it can be obtained from a valuable sample of blood. It can be from semen. It can be from skin. It can be from saliva. It can be from the follicles of hair. So again, this whole thing, I don't even want to underline a certain thing, but DNA is used for comparison and identification from multiple areas. But there's various uses of DNA. One is solving crimes, right? One is solving parental disputes, like the Maury Show. Used to determine the presence of genetic disorders, like DNA can let you know, hey, this runs in your family. As well as we want to keep track of different plants and animals. So you know how we track them with a piece of DNA, and then we have a little chip and stuff, and we can find them. That is what we use DNA for. But how does DNA analysis work? So you see this right here. This is the biggest thing that we're going to be learning about, which does answer a lot of questions. DNA fingerprinting is DNA. So again, this is not a fingerprint, though. Okay, so this is just a DNA pattern that is used by a technique called gel electrophoresis, which is this buddy right here. Okay. With gel electrophoresis, the idea is that the DNA is cut and the strands, obviously, smaller goes faster here, and which one we get down here, and we kind of just line up the strands. This is how we also able to find vaccinations if it works. This is how we're able to find um, who the baby daddy is. But the first thing is we got to remove DNA from the nucleus. So the first step is removal. Next thing is DNA will then be cut into smaller pieces by hundreds of enzymes. Each one cuts the DNA at a specific sequence in base pairs. So the base pairs, it could be every time it says CCC, we're cutting it at CCC, okay? So DNA is cut smaller. The thing is, we know that smaller things move. Like if I get a smaller ball, like a baseball, I can probably throw that farther than I can throw a football because of how big it is. So DNA has to be cut smaller. So removal, DNA is smaller, which is again manageable by enzymes. Then the gel pieces, the smaller pieces are now poured into the gel. Okay, and the gel is like is like a big old clear jello. And we have charges, just like in your car, we have a positive and a battery charge for you. And this is the same thing. So a negative charge is applied to the gel. The negative charge DNA is repelled. So we're going to start from the negative side and we move to the positive. So the next thing is, remember, we remove DNA. Oops, I did not. Sorry. We remove DNA. DNA is cut into smaller pieces. Then we put the gel. Then we go from negative to positive. And DNA is repelled and it starts moving to the positive end. Of course, the smaller pieces are going to move faster and further up the gel. The larger pieces will stay behind and take a long time to even get through the gel. Then the next thing is we compare the bands. And once we compare the bands, the idea is that we're looking for bands who are common or who are very similar. So the thing is, this is very, very particular. It cannot even be like a point something off. Here, if I give you a worksheet or if I give you an assignment to do, yes, it may have a little point something off, but literally, if you're really in a DNA analysis lab, it cannot. It has to be direct. Like I'm talking about this line right here. You see how that's a little bit crooked right here? No, that wouldn't even be a match. It would have to be like a direct perfect line, and I know I can't draw a direct perfect line. So this is basically what it is. So as we said with DNA, we take the strands and we take it. Once we get it, it's cut and we put it inside their gel electrophoresis. And so then it goes from negative to positive. As you see, it starts with kind of bigger bands, it goes to smaller bands or whatever, but smaller moves faster. So here is the process of making the gel. Okay, this is the process of getting the DNA. So we micropipette it into a tube. Once we get it into the tube, we have to make the gel, and we have to put the gel spacers in. Once we do this, it becomes here. This is the gel electrophoresis, as you see, is the nodes. As you see those blue lines right there are indicators of where DNA is at. So then it goes through the gel. Once it goes through the gel, it can be read. So here's an example of a cartoon version of what this would look like. So now we got to figure out, here is mom and here is dad. They got 4 kids. We got to figure out which kids are actually theirs by blood. So mom's DNA, dad's DNA. Now with this, because it's paternity, this is 50-50. Okay, so have to have 50 of mom's genes, 50 of dad's genes. All right, so let's look at the first child. Oh, this is a match. Oh, she's got dad's gene. Okay, we skip. Okay, this is dad's gene. And this, all right, that's obviously their baby. So we know that's mom and dad's baby. Let's look at daughter number 2. Hold on. This line ain't adding up to that line, and there's no line to match this line, but it's got mama DNA. Oh, this woman done had an affair, cheating on her man. That ain't right. That ain't his baby. Making him pay for somebody else's kid. Then they have a son. Okay, that DNA matches up. Okay, that one matches up. This one matches up, you know. This one matches up. This one kind of matches up. These lines are not perfect, but because it is a cartoon. But that's his baby. Okay. And now let's look at this one. Hold on. None of these matching up. He was adopted. Okay. But obviously mama was sleeping around because her middle child obviously got, okay. No questions asked. But that's how you read that for paternity. Now, let's say it's a crime scene and somebody killed the murder. This has to be 100% the same. Okay? So here is a blood stain. So now we got to figure out who killed who. Okay? So the blood stain that was found at the murder. So let's look at Bob's and see if his lines up. Well, he has 1, he has 2. And it's about, like, he doesn't have a lot, right? But it has to be 100% if it's your blood, okay? So let's look at Sue. Sue ain't lining up. John, he is basically identical. These are identical. That's how we do a crime scene. This is how we find out who someone did it. Their DNA matches up 100%. When people say, oh, we just getting their DNA fingerprint, we're getting a fingerprint. It's not just a fingerprint to get a crime. No, we got to get their DNA. Once it lines up like this, oh, we know who the person is. Human Genome Project. So this is pretty much, let me just break the word down. So in 1990, the U.S. and other countries began a Human Genome Project. The idea is analyzing human DNA. Human Gene Project. So we're taking human genes to obviously make sure we can get a clue that's to help us in life, such as making so all of this, yes, taking people's genes and lining them up. Idea is to do cure diseases and treatments for diseases, as well as, you know, just getting new vaccines. This is one reason why they're asking people who have COVID or who had COVID to donate their blood. So that they can do this, so we can get the best vaccination. All right, so gene therapy. So you know how we have physical therapy, we have a lot of different therapies, mental therapy, anything, I don't care. Gene therapy obviously is just for genes, but this is the process to changing the gene that causes a genetic disorder. So gene therapy, obviously we want to fix a misstate gene. Absent or faulty gene is replaced by a normal or working gene. So that's how one way we can do it. As well as oftentimes we're using a virus that has been embedded from a DNA containing normal gene into the patient. The virus will not make them sick, but it will deliver them the gene so that they can transcribe and translate new portions of DNA. You know what that is? When the doctors when you are born, you're given like 5 or 6 shots immediately. And those shots, our DNA is changed because that's not the DNA we was born with. See how it says here, it becomes a new portion of the DNA? Well, with that being a new portion of the DNA, what happens is when we get the flu shot, now we have the flu, yes, inside of us, the DNA of the virus. Well, the virus don't really have DNA, but we have, if you want to say it that way, that's the best way to say it, the DNA of the virus to come into our DNA and add to our DNA so that we can fight off the virus. So anytime you do get sick and stuff, your body does begin to make DNA fragments of that sickness, so you can not really get sick like that that way. But because things get mutated, things can change. Gene therapy. So again, we use gene therapy for people with SCIDS and cystic fibrosis. SCIDS is basically a way of somebody born with an immune system deficiency, and it's very severe. It's not on the level like AIDS, but AIDS is autoimmune disease. It's basically a deficiency again in the immune system for AIDS, right? So this is immune deficiency disorder, and cystic fibrosis is basically having overdue mucus. So gene therapies basically are for high risk patients and always are not always successful or not always long lasting. But the idea is to kind of give you something new, and hopefully your body will take. Then we have applied genetics. For thousands of years, everybody wants to make the best plants, the best animals, because like honestly, the bananas that we eat are mutated. The real banana is supposed to be covered like seeds, almost look like a papaya or something, and you kind of got to eat around, or like a dragon fruit. You see how it has all those seeds on the inside. A banana was like that, but they said the banana has so much potassium, so they actually end up making the banana not have seeds. That's why when you cut a banana and it has the black dots, because the seeds have been removed. So seedless grapes, seedless watermelon, yeah, that's stuff that we made for faster. Also, we do this for vegetables that are resistant to disease, and cows can produce more milk. Like, look at this. That ain't normal. There's 2 ways that we make this possible. We select the organisms and pick the type of phenotype. The phenotype is again physical appearance that we will want them to have, and based on this, we extract and manipulate the DNA. So first thing is selective breeding. So selective breeding, we can get it, just desired traits and make it from the parents. There are 2 types. We have selective, which is inbreeding, and we have hybridization. So like even the Shasta daisy, that's an example of selective breeding. Also here, inbreeding. So inbreeding is crossing 2 individuals with similar sets of specific traits. It's going to cause a mutation. Now we have an albino squirrel because of it mixed in probably with a sister or brother when they had babies. So when you have incest or interbreeding, that's basically the same thing. There's a genetic mutation, so it's too much. Definitely going to lead to that. Many genetic disorders. And hybridization, crossing 2 genetically different people and making something like this right here is not real. We made this. This is broccolini. Yes, that's why it looks like a lot of broccoli on the thing and then it has leaves on it. No, broccoli don't grow like that. Broccoli grow like a big old tree and we cut it down. This right here look like a whole bush. This is broccolini. This is something we made. We took 2 plants and mixed their genes together and they started having babies. Okay, so genetic engineering. So GMOs. So GMOs is genetically modified organism. Okay. And of course, please look at your stuff. Even though it says non-GMO, does not mean it's not non-GMO. You have to look at all the ingredients. So genes from one organism is transferred to the DNA of another organism. Okay. This DNA is now called recombinant DNA. This is where genetic engineering comes into it because everybody has recombinant DNA. The reason why? It's because you're always given shots when you was born. So we all have different DNA inside of our body. But this also helps produce medicines, improves food crops, helps human diseases and disorders. So like insulin and also like the growth hormone and the factor 8. Then we have stem cell research. Stem cells. I talked to you before about stem cells. Stem cells is a cell that becomes any other cell that both an adult and embryonic can be culturally grown in differentiate into different other specialized cells because it can, wherever it's needed, it will go. So the treatment of this is macular degeneration, diabetes, blood cancer diseases, and Lou Gehrig's disease. So cell transformation. A cell will take DNA from the environment, from the outside of the cell, and combine it with its own DNA. So this is how scientists can transform bacteria, plants, and animals, because we can take their DNA, put it in ours, make our DNA a little bit longer. Example, the isolated DNA that codes for production of insulin is joined with circular DNA plasma found in bacteria. So in order for us to get this insulin, we actually grow bacteria in a petri dish, which is real small, and I put a picture on it on the notes, and it produces insulin that can be harvested inside humans. So if you have diabetes, you got to take the false insulin. It's because something that we made, but it's beneficial to your body. There's different vocabulary for the genetics. We have vectors, which is vehicles. Okay, vectors, vehicles, and it's attached to transfer. What does a vehicle do? Transfer, drive you around. Foreign DNA into a new organism. So a vector is a vehicle to get the DNA, the foreign DNA to the new organism. We have biological vectors. So biological, which is viruses and stuff, and then we also have mechanical, which is micropipetting and tiny metal bullets. So applying genetics engineering, we have transgenic organism. Transgenic organism is somebody who contains foreign DNA, usually from another species. All right, but transgenic, what does that mean? Trans means change. Genic means genes. So if you hear someone say I'm transgender, that means, hey, I changed who I am, my sex, my gender. But if you are transgenic, you changed your genes. And when you change your genes, it's because you added in foreign DNA. All of us, honestly, is transgenic because we all have had, in one time in our life, a flu shot. We have now been giving a foreign DNA. We also all had different shots when we was born. It's like, especially the new generation. I believe when my daughter was born, on that same day, they gave her like 5 to 7 shots. So basis of the biotechnology, this is how the world is changing. Transgenic bacteria produces substances useful for health and industry. So again, we already talked about insulin, the human growth hormone, clotting factor. Transgenic animals are used to improve food supply. Look at this. This is a featherless chicken. So they're trying to say, oh, it improves it because we can get more chickens because it take a long time to pluck the feathers. So what they do is, hey, if we already have them born without feathers, we could just go ahead and cook them and throw them in there. That you got to think about that. Growing faster and leaner meat, when they get those cows being so big, you inject them with steroids. It's not really leaner meat. Transgenic plants supposed to improve food supply. Like for instance, like insecticide, just because we're trying to save the plants from not dying, but at the end of the day, if an insect bite that plant, they die. Eventually, if we keep eating that plant, what we're gonna do? I don't care if it's a plant or not. Those cells get in our bodies. So cloning. So cloning is when we manipulate DNA. Cloning is not that we try to get somebody in the machine and we make them, oh my goodness, boom, pop up here. No, these are genetically identical to the organism from which it's produced. So there's some ethical issues inside human genetics. So it's stem cell research, gene therapy, GMOs, cloning. Sometimes some humans don't agree with it because look at this. Because if we do something wrong, look at this right here. Yes, we call him the spider sheep. Look at him. That's crazy. Like all them legs. What were we doing? Imagine that walking. Okay. So here are some questions. Should humans be conceived in vitro? Well, in vitro means a tube baby. So let's just say you cannot, as a woman, you cannot carry a baby, but they take your eggs during ovulation, and your husband, he has to ejaculate in the cup. They take it, they freeze it, and they put it inside of a tube, and the embryo begins to grow inside the tube, and then they implant it inside the woman's uterus. The thing is, you don't know if it's multiple babies or if it's just one baby, but it's called a tube baby. This is how people are able to have surrogates and stuff. Is it ethical to have a child to save a child? Is it ethical for you to have a baby to save another child, as in maybe your other child is sick, but you needed a baby who has the same DNA to help them? Is organically grown food really better for you? That's just a real question to ask, because really, if it's mutated, is it really still organic? Should genetics be used to improve humans? I mean, I think so, because if we didn't, we wouldn't have some of the people who, like with diabetes, not living because we was able to produce some insulin, as well as people who are not able to have, who have different diseases, are able to, get healed and cured. And should genetic engineer bacteria be released into the environment? Um, I don't think so, because it's really what happened for COVID-19. But I do think that in some cases, that if it's needed, we can do testing to see how it goes. And then here you can just pause the video and just watch and read this article. Other than that, we're done.